Okay, so I'm going to talk about monohybrid and dihybrid Punnett square crosses. I think these are kind of fun to do, so I thought I would draw this out. Okay, so let's just, for example, say we are talking about dogs here. So let's say, for every trait, there's 2 genes. So let's say our male dog, our dad, he has the dominant trait of short legs. So that's going to be a big S. And then he also carries a gene for tall legs, but that's the small s. That's a recessive gene. So this is recessive. This is the dominant. And then our female, or our mom, let's say she carries both of the genes are the recessive trait for the long legs. So how we would cross this here, this is the way I like to do it. I just like to make a square. So for monohybrid, we're just going to have these 4 squares, and we're going to put the dad up top. It doesn't actually matter as long as you get if the mom is up here or the dad is up here, as long as you have the correct alleles, then it'll work out. So I guess this is kind of a bad example with the S's, but I'll try to make these look smaller. So now what we're going to do is just match up each S. So we have the big S, small s, small s, small s, small s, big S, small s, small s. You could also make it like this just so you can kind of tell a little bit easier. Okay, so when we look now at our phenotypes for the type of offspring we're going to have, so phenotype is like what we see. So as you can see here, 50% of our offspring is going to have long legs because they both have the recessive gene. And 50% of our offspring is going to have short legs because they have the dominant gene. So it doesn't matter that they have the recessive, the dominant always takes over the recessive. Oops. So that's how you do the monohybrid. That just means we're looking at one specific trait. When we do the dihybrid cross, it gets a little more complicated. Let's do a different example now. We'll stick with dogs, but what we'll do is, let's say the male has the dominant gene of spots, which is, so he has a big S and a little s, and then he also has, a pink tongue, and that's a dominant trait, and he has 2 of those. He doesn't have a recessive non-pink tongue. I don't know what the other thing we would say there is, but let's say our female has the recessive for spots, so she does not have spots, and she has the recessive tongue color. So let's say the dominant tongue color is pink and the recessive tongue color is brown. So now when we cross these 2, oh, that's an ugly square. It's going to get a little more complicated. So we're going to have 4 boxes across and 4 going down. So we're going to have 16 boxes. These are not equal, so don't judge my drawing skills, please. So how we're going to do this is match one of the first traits with the second one. So we're going to have big S, big P, then we're going to go here again, big S, big P, and then this S to this P, and again that S to that P. And then down here, so that's the dad, and then down here on the other side, so I'm just going to move this, we're going to have all of these are going to be the same because the mom holds the recessive genes for both of these. So SP, 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 SP. And now we're gonna cross. So I'm just gonna go through this kind of quick. That's a big S. I should have done better letters, but you know, oopsies. S, S, P, P, S, S, P, P, S, S, P, P. Okay, so now we're going to look at the possible phenotypes that we could get from the crossing of these two dogs. So we have, so if there's a big S, that means they're going to have spots, and if there's a big P, they're going to have a pink tongue. So spots and pink, and we'll go through and just highlight spots pink, spots pink, spots pink. Wow, that's a tongue twister. All of these have at least one big S and at least one big P. So 8 out of 16, so half of our offspring can have that. Now we're going to have no spots and pink, so we're looking for a big P, small S. Okay, so that's the other half. So I did not make this very creative. We could have done like a different setup here, which would have made a lot more variety in the types of offspring. So that's the phenotypes, and then the genotypes. Same thing. It's just if it has a big S or a big P is spots and pink, and a small S, big P, spots and pink. So that is pretty much how this works. Hopefully that made sense. Hopefully this wasn't too long. But yeah, essentially we're just looking at different traits, and the more we're looking at, the bigger the Punnett square gets. But yep, hopefully that makes sense. Thanks, guys.